The goal was to alter the water environment around the root and see how the pattern of root branches was affected. So why did I choose to work with maize, which is not necessarily the easiest organism to work with? So when I came into the lab, by and large, most of the researchers at the time were using Arabidopsis. It's in the mustard family. And that species is really advantageous for a lot of genetic experiments because it is very small, which means that you can grow a whole lot of plants in one greenhouse and do very large-scale experiments. But when it comes to looking at root development, the roots are very, very thin. They're only 100 microns thick, which means that if you want to look at what direction a branch is emerging around that circumference, it's very, very difficult. So there you do need to have a microscope to actually look at the roots, and it's a lot more laborious and time-consuming to work with Arabidopsis for this particular phenotype. But when I came in, Jose, my PI, was strongly encouraging of me trying to use maize as an alternative model. And the reason for that was because maize has these 1 millimeter thick roots, which make them very easy to work with when you're looking at environmental perturbations that vary across the circumference. So it's a lot easier to physically manipulate a larger, bulkier root like that than a really small, thin root like Arabidopsis. You might ask the question, like, why would you want to use maize specifically? You know, it has a large root, but there are other species that also have large roots. You know, there's rice and other larger species in the cereal crop family. Why not use those? And that kind of came down more to kind of the economics of things. So they say in America that corn is king. So that was a very strong impetus for us to use maize specifically because we saw it as being more immediately relevant to agriculture in the country that we were working in, thus making it potentially easier to get funding. And there was also a larger community of maize researchers that we could then interact with. So there were a lot of other benefits to using maize beyond just its role within the lab.